Alrighty, let me just get set up here. Okay. All right, thank you for joining me today. My name is Bridie Huckestein, and I will be talking about how we're trying to target metabolism to improve outcomes following severe influenza infection. I am a third-year PhD candidate in the Alcorn lab. So influenza has plagued human history for centuries. We first isolated the virus in the early 1930s, and since then we've tried to vaccinate against it. The first vaccine was made in chicken eggs in the 40s, and many of our doses are still made this way. However, cell-based vaccines are now being widely disseminated. Depending on the season, vaccines can vary in their effectiveness, and people are still getting influenza infections, creating a high burden on the healthcare system during flu season. The fact that there is a period of time named the flu season should tell you that it's still a prevalent problem. There are a few treatments available after you've been infected, like neuraminidase inhibitors, but they must be taken during the acute stage of the disease to be effective. A few days after onset of symptoms, many people will wait it out and miss this window in which current treatments options are effective. Even people who recover from influenza infections can suffer from lung impairment for several months to a year following an infection. For this reason, there is a huge need for more therapeutic options to improve outcomes for patients suffering from the long-term impacts of influenza. So while the virus itself causes damage to the lung epithelium, the immune response can cause just as much harm. Influenza elicits robust activation of interferon signaling and inflammasome activation, leading to the overproduction of cytokines or cytokine storm. This tips the balance of a protective immunity towards a pathogenic immune response or immunopathology. Here you can see x-rays taken from H1N1 patients on the left several days to weeks after infection, showing continued presence of cellular infiltration and inflammation. On the right are lung samples from the Anathi lab taken during autopsy that show increased collagen deposition and increased smooth muscle actin, indicating a fibrotic phenotype. So we use black 6 mice to model severe influenza infection and lung damage. In our model, viral replication, which takes place between 0 and 10 days, triggers the innate response, which is primarily driven by neutrophils, macrophages, NK cells, and dendritic cells, which then activate the adaptive immune response. You will find CD4 and CD8 T cells in the lung as early as day 4, while the antibody response doesn't begin until day 14. By day 11, once the virus has been controlled, lineage-negative epithelial progenitor cells, or LNEPs, begin migrating to areas of damage where they proliferate and differentiate into mature alveolar type 2 cells, which I'll get into more detail in just a minute. I just want to highlight here as well that any treatment I talk about will begin at day 14, post-viral clearance, and a few days after LNEP proliferation has begun. So the lower respiratory tract is composed of the trachea, bronchi, and lungs. The large airways, being the trachea and bronchi, branch off into smaller airways referred to as bronchioles, which end in sac-like structures called alveoli. The alveoli are responsible for gas exchange and are primarily composed of alveolar type 1 cells. These are the ones that do the gas exchange. A smaller percentage of alveolar type 2 cells are also present in the alveoli, which act as the sentinel of the alveolar compartment. So this on the right is from an older paper, and a lot of this is still being debated today, but there are many types of epithelial cells in the lung. They have distinct and some shared stem cell populations. In general, it's thought that basal cells and club cells give rise to club cells and ciliated cells and goblet cells. There's evidence that club cells can also give rise to alveolar type 2 cells, but following influenza infection, it seems lineage-negative epithelial progenitors, or LNEPs, are responsible for this. But again, this topic is heavily debated. It is generally accepted, though, that alveolar type 2 cells are responsible for repopulating the alveolar compartment, including both AT1 and AT2 pools. So the lung damage caused by influenza induces epithelial metaplasia that you can see here on the left. So the left, there are H&E slides from our lab showing the accumulation of non-immune cells where the alveolar epithelium should be. Compare this to mock-infected mice, and you'll see the lungs should look very airy and open. The Chapman lab stained these cells for mature epithelial lineage markers looking for club cells and AT2 cells, and found that the cells within these areas were lineage negative, but stained positively for KRT5 and could be identified by high levels of Notch signaling. Furthermore, they identified that compared to bleomycin-induced injury, KRT5 cells were failing to differentiate into mature cell types after flu, leading to reduced lung function. When we infect our mice with around 50 PFU of PR8, the mouse-adapted H1N1 strain of influenza, inflammation is present in the lungs for at least 35 days and often longer. I ran a bioplex assay looking at several different pro-inflammatory cytokines in the lung 21 days post-infection and found significant increases in several, including TNF-alpha, IL-6, IL-12P40, and MCP1. So this is a very simple diagram to explain a very complex signaling pathway. The mammalian target of rapamycin complex 1, or mTORC1, is a central mediator of cellular metabolism, receiving input about the energetic status of the environment like oxygen levels, growth factors, and glucose, and when these things are in high availability, telling the cell it's okay to make proteins and lipids. So essentially, activating mTORC1 is going to increase anabolic metabolism and decrease catabolic metabolism. A study by Bin Huang et al. Looked at stem cells, particularly osteoblast differentiation and mTORC signaling. They found that as the osteoblast matured, mTORC1 activity decreased. They then discovered that mTORC1 was responsible for sustaining Notch signaling in osteoblasts, preventing their differentiation. This has been repeated in a few other stem cell types, but its role in LNEPs is unknown. If you remember, LNEPs were identified by having high Notch signaling, so we looked to see if they had high mTORC1 activation as well. To answer this, I performed flow cytometry to look at LNEP-specific mTORC1 activation. Using an antibody for phosphorylated ribosomal protein S6, I saw increased levels of activation in 2 separate experiments. I then looked at CD45 positive cells 7, 14, and 21 days post-infection to determine if treating with an mTORC1 inhibitor would also impact immune cells in the lung, but saw decreased phosphorylation over the time course. So it was likely that treatment would more heavily impact non-immune cells than immune cells. So to summarize what I've told you so far, I've told you that influenza causes long-term lung damage and inflammation, and that at least a portion of this lung damage is characterized by clusters of undifferentiated stem cells called LNEPs, and that these LNEPs have high mTORC1 signaling. So together, this data led me to believe that mTORC1 activation in LNEPs causes epithelial metaplasia, delaying the repair process and contributing to the inflammatory phenotype we see. So to address this, I infected 2 groups of mice with the mouse-adapted strain of influenza, PR8, and then treated 1 group with the mTORC1 inhibitor rapamycin 14 to 20 days post-infection by oral gavage. This time frame was chosen because it was after viral replication had ceased and also gave LNAPs a few days to proliferate before inhibiting mTORC1. I'm currently analyzing several different endpoints to determine how this treatment impacts alveolar stem cell differentiation, lung pathology, and inflammation, including histology, qPCR, cytokine analysis, and lung leak. So I took pictures of my lung slices at 4x and then used Photoshop to stitch them together. So please excuse the poorly stitched together image, but just to get a global view of what these lungs look like. The pathology scoring was done on the microscope, and interestingly, there seems to be a small trend indicating that rapamycin actually made the damage and metaplasia worse. But now I need larger sample sizes to determine if this is significant. I also did a large flow cytometry experiment where I infected mice with PBS or PR8, which would be my flu controls, and then treated flu-infected mice with rapamycin 14 to 20 days. So those are my RAPA group. I ran both lung and bowel samples from myeloid and lymphoid panels, and the bowel mirrored what I saw in the lung. So I'm just going to show you the lung data. Overall, there were no differences in CD45 positive cells, B cells, or T cells with treatment, but the dendritic cell compartment was affected. We saw a significant increase in classical dendritic cells in the RAPA-treated group, as well as a decrease in the non-classical DCs, which primarily produce interferon gamma. And we also saw a decrease in Ly6C high monocytes. Additionally, there were no differences in any macrophage or NK cell populations. So going forward, I will increase my sample sizes to see if the pathology that we see trending is real. It is likely that rapamycin impacts the DC compartment, so it'd be interesting to see if this is what's making the pathology worse. I still have a lot of experiments to do, so I will perform cytokine analysis on lung homogenate, use immunohistochemistry to look for alveolar type 2 to KRT5 ratios, and try a time course to figure out if there's a good time for treatment. And with that, I just want to say thank you to my amazing lab, Dr. Alcorn, Danny for helping me with all of my experiments, Elise for making these panels, and I'm sorry your name is spelled wrong on there, my undergraduates for doing a lot of qPCR, my thesis committee for constantly giving me great advice, and the Anathi lab for the histology. Thank you. Have a good one.